Okay, so I'm going to start to introduce you. So good morning or good afternoon to everybody. I hope you hear me well. So today the webinar is going to be given by Dr. Jean-Pierre Valentin. So on cardiovascular toxicity assessment in pharmaceutical drug development. As you know, it's very important during the development of any kind of pharmaceutical compound. And so Jean-Pierre is a pharmaceutical scientist with over 30 years of experience in the biopharmaceuticals industry, spanning both scientific and senior leadership roles across several companies. He has contributed to the discovery of numerous drug candidates and has overseen the safety and secondary pharmacology assessment of approximately 200 compounds across multiple therapeutic areas. So actually, Dr. Jean-Pierre Valentin is a senior director, head of investigative toxicology at UCB Pharma, and is a former president of the Safety Pharmacology Society, and he currently holds leadership roles within HESI, IQ Drug Safe, and FPA PDAG. He also represented EFPIA, on the ICH E14S7B, which is the guideline on safety pharmacology implementation working group, and has been actively involved in global regulatory science initiatives. So he has a long-standing commitment to training, I can say, because he's giving courses in our master program and mentoring through scientific societies, universities, and trade associations, and he's the author of several patents and more than 250 scientific publications. So, as you can see, Jean-Pierre is a great expert in the field of cardiovascular toxicity. So please turn off your microphone. And as you know, Jean-Pierre is going to give you the talk for 45 minutes, and the last 15 minutes I will read the questions to the chat. So please put the question in the chat. So Jean-Pierre, thank you very much again for giving this presentation, and the floor is yours.
Thank you, Mark, and good morning, good afternoon, everyone, and thank you very much to the EUROTOX for the invitation and opportunity to present today. So as Mark said, the topic of this webinar is on cardiovascular toxicity assessment in pharmaceutical drug development. So today I will provide a strategic overview of how cardiovascular safety is being assessed, how methodologies have evolved, and how emerging technologies may shape the future in this field. So first, let me briefly outline the structure of this presentation. First, we will examine the importance of cardiovascular events across the pharmaceutical life cycle. Then we will discuss key elements required to design a cardiovascular toxicity strategy. Next, I will review a typical non-clinical cardiovascular assessment approaches, including in vitro, in vivo, and computational tools as well. We will then look at drug-induced QT prolongation as a regulatory success story, followed by emerging regulatory thinking around blood pressure effects. And finally, I will present several case studies before concluding. So cardiovascular safety remains one of the most critical challenges in drug development. It influences decision-making throughout the entire life cycle of a drug, from very early in drug discovery to clinical development and even post-approval pharmacovigilance. So cardiovascular safety issue can arise at every stage of the drug development life cycle, as illustrated on this slide. During early discovery and development, cardiovascular toxicity contributes significantly to candidate drug attrition. Later in drug development, cardiovascular events can cause clinical trial delays, program termination, or regulatory restrictions. Even after approval, cardiovascular adverse drug reaction remain among the most common serious and drug-related safety issues. So this really highlights why cardiovascular safety evaluation plays such a central role in modern drug development. Cardiovascular findings are frequently observed during non-clinical studies, as illustrated on this slide. Large dataset from telemetry studies and toxicology studies show that compounds often produce effect on heart rate, blood pressure, ventricular contractility, and the electrocardiogram, such as the QT interval. In addition, histopathological findings affecting myocardial tissue or cardiac valve may be detected during repeat dose toxicology studies in a significant proportion. This observation illustrates the fact that cardiovascular safety signals are relatively common in a non-clinical setting, and therefore they require careful identification, monitoring, interpretation, and contextualization. Cardiovascular adverse events also occur during clinical development and even post-marketing surveillance. Analysis of regulatory safety databases, such as the FARS database from the FDA, show that cardiovascular events such as arrhythmia, hypertension, myocardial injury, and vascular complication remain among the most frequently reported serious adverse drug reactions. So therefore, and again, improving prediction and mitigation of cardiovascular risk remain a major priority, not only for us in the industry, but also for regulators, because ultimately it may impact patients' life. So the consequences of cardiovascular safety liability can be very profound. Several high-profile cases illustrate this point, and some are captured on this slide, but certainly not all of them. Drugs such as terfenadine, rosiglitazone, and benfluorex were either withdrawn from the market or subject to major regulatory restriction because of cardiovascular safety concerns. Other compounds have required large cardiovascular outcome trials or additional risk management and mitigation programs. These examples again demonstrate that cardiovascular safety is not just about drug attrition or discontinuation, but has a major implication for patients ultimately that are taking these drugs, regulators, and pharmaceutical companies alike. So now changing subject, it is clear that drug-induced cardiovascular toxicity can affect multiple components of the cardiovascular system and can be structural and or functional in nature. And these include electrophysiological effect, hemodynamic changes, myocardial injury, vascular dysfunction, or structural cardiac alteration. Such effect may occur acutely or develop after chronic drug exposure. So the diversity of possible mechanisms makes cardiovascular safety assessment particularly challenging. It also explains why multiple complementary models and approaches are required. One additional element is nowadays the diversity of novel therapeutic modalities that add an extra layer of complexity. So illustrated on this slide are the reference and some examples that I will present later, such as the check inhibitor, the plasmapsin 10 program. So where we stand today. So some recent work has attempted to categorize cardiovascular toxicant according to key mechanistic characteristics.
Jean-Pierre, there's an issue for me on the slide because it's so. Is it the same for the others?
No? Yes.
So where are you?
Superimposing. Yes, that's good. That's okay. This way it's okay.
So do you
now
see
it's okay.
Okay. I haven't touched anything, but I will progress and let me know if there is any other issue.
Okay.
Okay. So as I was saying, so recent work has attempted to categorize the cardiovascular toxicants according to their key mechanistic characteristic. These include processes such as oxidative stress, mitochondrial dysfunction, inflammatory signaling, and channel disruption, or impaired vascular regulation. So clearly identifying these mechanisms helps to guide the development of assays that are capable of detecting relevant hazard. It also facilitates the development of adverse outcome pathways linking molecular events to clinical outcome. So can you see the slide clearly, just to confirm?
I think there is a slow transition, you see, because again, it is superimposed.
Okay, so it may take a few seconds.
Yeah.
Okay.
Right now, it is still. It's not moving. I mean, the letter one is superimposing with the next one.
Okay, so.
Yes, that's okay now. So it takes some time.
Okay, apologize for that. I'm not sure I can do anything about it. It's probably the network. So on this slide is illustrated the fact that a broad range of models is used to study cardiovascular safety. These range from simple molecular assays to complex in vivo studies. Each system provides, in effect, different types of information. Some assays offer high throughput but limited physiological relevance. Other assays provide better representation of human physiology but have a lower throughput. So clearly one of the central challenge in safety testing is therefore balancing the throughput, including compound requirement, mechanistic insight, and translational value. So I've changed slide, so I will wait a couple of seconds. This slide illustrates the fact that the models used for cardiovascular safety assessment continue to evolve over time. Some new ones are appearing all the time, whereas some others are being sunset, okay, because they are superseded by newer, more reliable or more predictive models. So new technologies are regularly being introduced into drug discovery pipelines. And however, the regulatory guidance often develop more slowly, and it may take several years before a new guidance is being introduced. As a result, industry frequently adopts new approaches well before regulatory guidance exists. This dynamic environment requires, in effect, continuous scientific evaluation by ourselves as well as service providers, technology developers, and ultimately regulators. So again, I'm changing slides, so a couple of seconds, hopefully for the slide to materialize. This slide illustrates the fact that
maybe you should click twice on to pass them.
What if I click
yes,
twice?
That's okay. If you click twice, maybe it could be faster.
Okay, but I need to come back then, because if I press twice.
Okay. So now it's okay. Now it's okay.
Okay. So an important question when developing an essay and interpreting cardiovascular data is determining what constitutes, in effect, a clinically meaningful change. So small variation in a given endpoint or parameter, such as heart rate or blood pressure, may not translate into clinical risk, although we acknowledge that some small change in blood pressure or heart rate may have some long-lasting significant clinical implications. So understanding the relationship between non-clinical signal and clinical outcome is therefore critical. This requires integration of pharmacology, exposure relationship, and clinical experience. One example captured on the first row is around QT prolongation, for which the threshold of scientific and regulatory concerns are different. 10 milliseconds being considered as a threshold of regulatory concern, whereas clinically speaking, a 40 to 60 millisecond change is being considered as a threshold of clinical concern. So I'm pressing twice and coming back. So I hope you can see a slide titled.
Excellent.
Okay, so I will try to juggle as we go along. So when implementing new assay technology, it is essential to build the confidence in their predictive value and predictive capacity. So typically there are three main factors that are typically considered. First, it's about the biological relevance, meaning how well the model reflects human physiology or physiopathology. Second is the assay robustness and reproducibility within and between laboratories. And the third element is what we define as human translation, human relevance, which is often evaluated through pharmacological validation using a set of compound reference drugs. So these factors, in effect, help determine whether a model is fit for its intended purpose. So I press twice and go back once. Okay. So on this slide, you can see kind of cardiovascular safety assessment paradigm. And we need to consider all its components in a broader context of risk assessment, risk management during all stages of drug development. Early in discovery, the goal is often to identify hazard and eliminate the problematic compounds. Later in development, when the compound progresses into clinical development, the emphasis shifts towards quantitative risk assessment, integrating non-clinical as well as clinical data as they emerge. This evolving framework that is in effect context of use dependent ensure that safety evaluation remains aligned with the stage of drug development. And at the bottom of this slide, the assessment of drug-induced QT prolongation is used as an illustrative example with all the different types of assays that are being conducted preclinically and clinically throughout drug development. So I press twice and back once. So this slide presents a typical cardiovascular safety assessment strategy, and it integrates multiple experimental approaches. These include in vitro assay, in silico modeling, in vivo telemetry studies, toxicology studies, and ultimately clinical monitoring when a compound will progress in the clinic. Each of these components contributes to a comprehensive evaluation of cardiovascular risk. Importantly, these data are interpreted using an integrated risk assessment approaches rather than relying on a single individual test system. That said, there are some binary decision type go, no-go that are being made based on some of the most robust models for screening purposes, such as, for example, the ERG assay. And talking about the ERG assay, I press twice, come back. In the next few slides, I will describe the different assays that are being deployed in drug development and at which stage they are being used. So in terms of ion channel screening, it's really a cornerstone of cardiac safety pharmacology. Electrophysiological assay evaluate the effect of compound on key cardiac ion channels such as ERG, NAV 1.5, CAV 1.2, and a few others. These assays provide quantitative measure of channel inhibition such as the drug concentration inhibiting the current by 50% or IC50. And it helps identifying compounds with potential electrophysiological liabilities. Some computational modeling are being used, and they are increasingly used to complement experimental studies. Some structure-activity relationship, or SAR, models can predict ion channel interaction even before compounds are being synthesized. This approach helps medicinal chemists to design safer molecules earlier in the discovery process. And again, ERG has been the perfect example of the success of such approach. There are also more advanced computational approaches that are being used, such as the one that simulates the human ventricular action potential. By integrating information from multiple ion channel IC50 data and other input, this model can predict how a compound may alter cardiac electrophysiology in either healthy population or disease patient population. And that's the spread of the population of action potential that you are seeing on the right-hand side. More recently, over the last decade, human-induced pluripotent stem cell-derived cardiomyocytes represent another important platform. These cells provide a human-based system for assessing electrophysiological effect, contractility, and cellular toxicity. Although these models still have some limitations, particularly related to the cellular maturity of these cells, they do offer valuable opportunity for improving translational and human relevance. Another limitation of these type of assay is that they represent a single cell type and therefore not mimicking the full human heart. Despite significant advances in vitro and in silico models, the in vivo telemetry studies remain a cornerstone of cardiovascular safety pharmacology. Telemetry allowed continuous monitoring of the electrocardiogram, blood pressure, and cardiac contractility in conscious, freely moving, group house animals. These studies provide an integrated physiological assessment with a high direct translational value to human. Recent technological advances have also introduced non-invasive telemetry systems that can be incorporated into repeat dose general toxicology studies. That's the main advantage of such approaches. This allowed cardiovascular monitoring under chronic dosing conditions and improve the integration between safety pharmacology and toxicology programs. An example of a compound inducing a progressive delay dose-dependent QTc prolongation is presented on this illustration. Hopefully you will see it shortly. And it's really to highlight the fact that such effect would have not been predicted in a standalone single-dose safety pharmacology study. So beyond these standard models that are used routinely, when safety signals are detected or suspected, additional follow-up studies may be required to understand or characterize the underlying mechanism. These can include, but not limited to, isolated heart model, imaging techniques, biomarker analysis, and mechanistic cellular or subcellular assay. The goal here is really to determine mechanism, and clinical relevance. So, changing gear here to focus on what we define as a success story. So it's about drug-induced QT prolongation as one of the most intensively studied cardiovascular safety issue over the last 25 years. Over the past 2 decades, regulatory frameworks have been developed to evaluate and mitigate the risk of torsades de pointes, the potentially life-threatening arrhythmias. Strategic and regulatory frameworks have evolved over years based on the increased scientific knowledge and mechanistic understanding of the issue, as reflected on the next slide. So, in effect, 2 key guidelines address this issue. The ICH-S7B focuses on non-clinical evaluation, whereas the ICH-E14 addresses clinical QT assessment. Together, they form a comprehensive regulatory framework that has been very impactful. Nowadays, over 80% of the clinical QT assessment studies are either negative or with very small effect of deemed of no safety concern. And this is really because of the hard and significant work that has been done in a non-clinical setting by deploying the strategies that I presented earlier that have enabled to progress compounds that are in effect do not carry ERG liability or QT liability in a clinical setting. So why would we continue to do these very thorough and expensive TQT studies? In effect, a recent update to the joint ICH-E14 and S7B has emphasized the value of the integrated risk assessment by combining multiple lines of evidence and, most importantly, by combining the non-clinical and the clinical evaluation. This integrated approach has significantly improved our confidence on the prediction of priority risk and optimize and streamline our drug development approach. So this slide illustrates how the regulatory strategy for evaluation of drug-induced QT prolongation has evolved in the past 2 decades. Historically, following the introduction of ICH-S7B and E14 around 2005, QT risk assessment was largely sequential and somewhat fragmented. Non-clinical studies such as the in vitro ERG-assay and the in vivo QT studies were performed to support entry into first-in-human clinical trials, but the clinical evaluation relied heavily, if not exclusively, on the thorough QT studies or TQT studies, which became the regulatory gold standard. Over time, however, it became clear that a more integrated approach could provide better scientific insight and potentially reduce the need for these dedicated TQT studies. So the latest update in 2022, so 4 years ago, introduced the concept of an integrated QT risk assessment where non-clinical and clinical data are evaluated together, improving regulatory decision-making and certainly, in many cases, streamlining drug development. So the cardiac electrophysiology data are then combined together to form an integrated risk assessment enabling the establishment of a margin of safety, which is a critical element in our decision-making process. And what's presented on this slide is what we call in our jargon a QTogram. Okay, so the integration of all the information, all the data that relate to QT prolongation. So the safety margin is typically calculated for each of these essay or endpoint, and it's typically calculated as a ratio between the key safety endpoint, for example, the ERG IC50, or 10 millisecond prolongation of the QT interval, against the free, i.e. the unbound therapeutic plasma concentration, which is either measured or anticipated or suspected when we do this exercise very early in drug discovery. Such assessment enabled to make informed decisions regarding the suitability to progress a compound into human clinical trial. Now, switching topics again, talking about blood pressure effects that have, over the recent years, received an increased regulatory attention. Recent discussions have considered whether systematic evaluation of blood pressure effects may become more common in clinical development. And some of the reasons are captured on this slide. So, it is well known that increased risk of cardiovascular serious adverse event, including mortality and morbidity, is associated or there is a causal relationship with increase in blood pressure, and some of these increase in blood pressure may be very small. Even tiny but chronic blood pressure elevation may have detrimental effect. So a revised draft clinical guidance has been issued by FDA a few years back to advise the sponsor on the pre-marketing assessment of drug effect on blood pressure. It is applicable to new drugs with systemic bioavailability, although with a clear emphasis on drugs intended for chronic treatment and a focus on best practices for clinicians. Interestingly, or I would say surprisingly, there is absolutely no reference whatsoever to non-clinical assessment in the guideline, which may appear some kind of missed opportunity. Why am I saying that? The reason is illustrated on the next slide. In effect, nowadays, there are some nice quantitative system pharmacology or QSP models that are emerging and that integrate the polypharmacology of a drug as well as the physiological component driving certain parameters such as blood pressure. And these models can simulate cardiovascular response and help to interpret complex safety signals such as drug effect on blood pressure. Such model integrated information from on-target and off-target profiling as well as the physiological driver for modulating this parameter. And all of this lead to a prediction of blood pressure effect. These are very promising tools, and it's unfortunate that they are not being considered as part of such guidance. Again, switching gear now, I will focus on 3 examples to illustrate a little bit what we do in real life. And the data from these 3 examples are available in the public domain. So this first example illustrates the effect of a compound associated with negative inotropic effects, and mechanistic studies were used to identify the potential underlying mechanism, including polypharmacology, and to assess the potential risk. So the result highlighted the polypharmacology contributing most probably to the effects that were consistently picked up by more complex models, in vitro isolated heart or in vivo, and they align, unfortunately, very closely with the clinical readout illustrated by the kind of diamond symbol. And unfortunately, in this case, the compound had to be discontinued from the clinical development. The second example referred to a drug that was associated with significant cardiovascular effects, in particular cardiac histopathology, that were strongly suspected to be linked to the hemodynamic alteration, namely sustained hypertension and tachycardia. In effect, preventing the tachycardia and the hypertension simultaneously fully prevented the pathological readout, namely the necrosis, and the elevation of cardiac biomarker, in effect, the troponin changes. So clearly this highlights that understanding the mechanism allowed to resume the clinical development in a kind of safe manner. The third example refer this time a case study involving QRS widening in a kind of anti-malarial discovery program. So some detailed mechanistic studies using molecular, cellular, and tissular in vitro assay identified common polypharmacology as the likely root cause, translating all the way through to in vivo assessment. So there was a nice relationship between in vitro and in vivo assessment. And this deconvolution enables the medicinal chemist to design out such liability at different cardiac ion channels and to develop compound with an acceptable safety margin against QRS widening. So looking ahead, cardiovascular safety assessment is becoming increasingly integrated. Advancing human-based models, computational approaches, biomarker, and artificial intelligence will continue to improve prediction of cardiovascular risk. At the same time, adoption of a new approach methodology and the 3R principle will influence how safety pharmacology evolves. Ultimately, cardiovascular safety is no longer a single test. It is integrated as a scientific discipline combining pharmacology, physiology, toxicology, and regulatory science. However, a word of caution presented on this slide, and this slide summarizes the data from a survey, the same survey taken 10 years apart. It highlights the transformative potential impact of NAMs and associated assay and technologies in safety testing, emphasizing that the field is rapidly evolving, but changes often unfold over several, if not many years. While some NAMs, technologies, and associated assay have been successfully integrated into mainstream drug safety assessments, such as, for example, high-content imaging or IPSCs, others face challenges in adoption, such as organ-on-a-chip, or some have not succeeded yet, such as microRNA or metabolomics. This evolution reflects the need to balance innovation with confidence in predictive performance. Ultimately, the goal of NAMs is not necessarily to replace all traditional models immediately, but rather to complement existing approaches with and improve the prediction of human safety outcome while supporting the 3R principles. Drug discovery and development are currently undergoing a significant paradigm shift. Historically, most pharmaceutical pipelines were dominated by small molecule, and many safety strategies were therefore developed around this modality. However, today, drug portfolios are increasingly diverse and include monoclonal antibodies, peptides, oligonucleotides, antibody-drug conjugate, and many other novel modalities. These different different therapeutic modalities interact with biological systems that are fundamentally different between them and from the original small molecule world. As a result, the traditional small molecule paradigm for safety assessment does no longer apply directly. This requires, therefore, to adapt our approaches to secondary safety pharmacology and toxicology testing so that they remain fit for purpose across a broader range of therapeutic modalities, including when it comes to cardiovascular safety testing. So moving on to the very last few slides. This slide illustrates the transition that is being underway in drug development. So traditionally, discovery rely on simple 2D in vitro model, followed by extensive animal testing in preclinical development before moving to patients. Nowadays, we are increasingly using more complex human relevant models such as organ-on-a-chip, and in silico approaches, alongside more refined animal studies. So now looking ahead of the next few years, if not decades, integrated human-based model and computational tools could substantially reduce reliance on animals, but reaching that goal will still require time, validation, and regulatory acceptance. So the message is, it's not for tomorrow. So that's my summary and conclusion. Clearly, cardiovascular safety remains a major focus through the entire drug development life cycle, as illustrated through this presentation. Over time, the field has evolved from relying primarily on individual assay toward a much more integrated translational approach to really integrate all components of the risk assessment. The experience gained from QT prolongation risk assessment demonstrate how mechanistic understanding, improving experimental model, and regulatory guidance can work together to reduce clinical risk to patients and streamline drug development. Today, the toolbox available to scientists is expanding rapidly and now include in vitro models, computational approaches, artificial intelligence, as I said, biomarkers, and advanced in vivo technologies. However, confidence in these approaches depend on the biological relevance, the assay robustness, and the human translation, and clearly define a context of use that's clearly of primary importance. So looking ahead, cardiovascular safety testing will continue to evolve through innovation, collaboration, and integration of emerging technologies. So last but not least, I would like to thank colleagues from many organizations who over the years and many discussions and exchanges have helped developing and maturing the topic presented today. So a big thank you to all of them. I will not mention them, but they will recognize themselves. With that, I would like to thank you all very much and apologize for the slide delay in moving the slide. Hopefully you got the key message from this slide. Thank you very much and happy to take any question.
Thank you very much, Jean-Pierre. Well, finally, it went well for the slide with a new way of passing it. So it was okay. New issue. So there is some question in the chat that I will read it to you. So first question from Nikolaos Kritikos. So what is the basis of your in silico models? Is it based on pure physico-chemical characteristic of substances, or are you using? Are you using? Are you using topological indices or even drug-like? Cannot read it. I'm sorry. Or drug-like indices. The later. Okay. Or you can start maybe with you.
Yeah. So in effect, there are a plethora of in silico models that are being developed and used. And I just presented a couple of them. First one about ion channel prediction of interaction potency at different target is based on a suggested physicochemical properties and chemical structure. It's really a structure-activity relationship type models that are being developed. And these models there are several of them, or many of them, I would say, that are either commercially available, or some are publicly available, and some are more proprietary. And the proprietary one have been in effect refined so that they are more focused on the chemistry of a given organization. And the second type of model that I presented is really providing different input, collecting different input in order to release an output. So in this case, it was capturing the IC50 and different parameters from different ion channels in order to predict an effect of a drug on the cardiac action potential. And again, there are several models available publicly, commercially, or proprietary. So again, it's a topic on its own, so I will stop there for now. I hope it addressed in part the question.
So second question. I mean, do you think that iPSC could eventually replace the ERG assay for comprehensive arrhythmic risk assessment?
That's a very good question. And it's true that some organizations are asking the question whether it's preferable to start first for assessing drug effect on different cardiac ion channel, or to start looking immediately at an integrated system that will give an overview of the effect on the cardiac action potential, and if an effect is seen, then to deconvolute back to the mechanism. So the two options are there. I'm aware of companies that are using one or the other. Okay, so it's already in place in certain organizations. It's really left down to the kind of strategic approach that any organization want to follow.
So for still completing this question, I mean, for example, could you use both systems in a way that the ERG assay, you can use it very early in the development for candidate selection or EAT to lead, and then use the cardiomyocyte iPSC later in the development? Is that yes. Possible? Or? Yeah.
Yes, absolutely. So as I said, the two approaches can be used. So starting with kind of early screen that have a high throughput and low compound requirements, such as ERG, NAV, CAV, assay, and later on a more integrated system to confirm typically the absence of an effect on the at a cellular level, or the other way around, an integrated system first, which come at a cost because it's more costly, etc. But by not doing the ion channel assay, it offsets the cost. So it's really down to a strategic decision of an organization.
Okay. So now, is it accurate to say that drugs having an activity adverse effect on the cardiovascular system have a partial affinity in known targets of the system, or is it more typical to see a more complex effect that goes as far as affecting transcription, for example, affecting protein transcription?
I will need to refresh my memory back to some review that analyze the compound that were discontinued, withdraw from the market, as well as ones that failed in early or late clinical development. I would say intuitively, I would think that it's a combination of both. Some have, and it's in part the message that came out from the paper from Lind et al. From 2021 that I presented One's about the key characteristics, and you can see there was a plethora of physiological processes that were covered out of these 12, and therefore underpinning the fact that some effects were direct, some indirect, some simple interaction, some much more complex, with leading to AOPs, etc. So it's a combination of everything, which make, as I said, the assessment of cardiovascular risk a real challenge nowadays in our industry.
So I still have some questions. So in cases where you have employed both animal testing and QSAR and in vitro testing, how frequent has it been to have an animal experiment contradict the results of in vitro QSAR?
I'm not sure that's the way I would phrase the question, because ultimately what we are trying to predict is a clinical outcome, first and foremost. But acknowledging that at this moment in time, we still have to go through the in vivo experiment. So there are instances where effectively none of the model have a 100% predictive capacity in terms of sensitivity, specificity, positive and negative predictive value. So none of them have this power, but it's a question whether it's because the endpoint being an in vivo animal study is the right one, or is not, because we are trying to predict the human situation. It's probably a bit of both. In some instances, the model simply don't recapitulate either the full animal physiology or pathophysiology or the human physiology or pathophysiology. So we still have work to do there. And this is what, just to finish on this question, it's important nowadays not to rely necessarily on one model, but to integrate multiple models in order to form an holistic view on a given problem.
So more questions. So when would you expect that cardiac organoids and spheroids will become used in vitro preclinical screenings?
That's a difficult question. That's really a difficult one. I think it's, I can make several points here. First one is it's really an internal strategic decision. So each company, whether it's a large pharma, mid-sized pharma, or small biotech, would have a different perspective on this question. Another aspect is typically industry is very reactive when the regulatory guidance is telling us what to do. So for example, in this specific case, if there was tomorrow a guidance telling us that we have to use some complex cardiac in vitro model, the industry would comply. Okay. I'm not saying whether this would be the right thing to do or not. I'm just stating some facts here. Now, I think all in all, it will depend on the confidence that any given scientist would have in the model. The context of use, is it for screening purposes? Is it for investigative purposes? Is it to support some regulatory claim, etc., etc.? So all these factors will be taken into account, I believe, by any given organization. So it's not a clear answer, okay, but it's just set up the scene.
Okay, so I've got one more question. So based on the FDA blood pressure guideline, we understand that increases in blood pressure are a sensitive topic. Is there any guidance from regulatory authorities on threshold levels or criteria to evaluate blood pressure effect in non-rodent studies, or will the current routine approach remain appropriate?
I think again, it's a very important question and critical question. I think what was disappointing, at least that's my opinion, disappointing with this FDA draft guidance is that it doesn't leave any space, any room for non-clinical evaluation. So irrespective of the non-clinical evaluation, a thorough clinical monitoring of blood pressure would have to take place, again, under certain circumstances for certain compound, etc., but nevertheless, there is no room for a non-clinical evaluation. So basically, irrespective of what the sponsor has done, it will not impact the clinical assessment. A relief for now is that this guidance is still in its draft stage and has not been promulgated as a final guidance. So we are hopeful as an industry that there will be possibility to engage and continue the engagement with FDA to move more toward a situation as we have experienced with QT, being able to integrate non-clinical and clinical data.
Okay, so we have no more questions in the chat. And we are approaching the end of the presentation because it's nearly 2 o'clock. So Jean-Pierre, we have many questions. So I think people are very maybe one more question. No, I've got many thank you in the chat. So it has been well appreciated by the public. So thank you again for the presentation. It was very comprehensive. Lot of questions, lot of good answers. So I think everybody is aware that cardio safety is an important issue, and there is still some stuff to do. Okay. So thank you again, Jean-Pierre. It was very interesting. So thank you.
Thank you very much for the invitation, and happy to take any question outside this forum. Thank you. Bye-bye.
Thank you. Bye-bye, Jean-Pierre.